Hello and welcome to my presentation on Microbiology and Me. I'll start by introducing myself. I am Jade, a STEM ambassador, and I'm currently an industrial placement student at GlaxoSmithKline in Pharmaceutical Microbiology, which I will discuss more later on. This presentation will give you an insight into my journey through education to where I am now, including work experience, the university I am at, my placement/my role, and any advice that I have. At GCSE, I always had a real passion for science, but I wasn't sure where I wanted to go with it in terms of a future career. I knew I wanted to go to university, and when applying for university, they have requirements for grades as well as subject-specific requirements. So I wanted to keep my options open and decided to pick biology and chemistry as 2 out of the 3 of my A levels, as well as psychology. Through this, I found the field of biomedical science, and this motivated me to seek some form of work experience so that I could see for myself what it means to work within that field of research. I was fortunate enough to secure 2 hospital opportunities at Birmingham Children's Hospital and Queen Elizabeth Hospital, where I saw firsthand the wide range of sectors encompassed within biomedical science. From working in clinical labs, I was able to go into various departments such as histopathology, which is the study of diseases of tissues, hematology, which involves working with blood diseases and cells. But ultimately, it was the work I was able to do in the microbiology department, which inspired me to pursue a degree in that field. Being able to see how hands-on the processes were and the level of problem solving required to choose each test we needed to carry out in order to diagnose a problem was a light bulb moment for me. I was proactive in learning more about microbiology and what it could entail, so I began some online courses through FutureLearn. I took online courses in biochemistry, which looked at the molecular techniques used in microbiology, such as mass spectrometry. I also took courses in histology, which looked at infections in tissue samples. I was able to research more into microbiology and the possible areas within the subject, but it was the online course in antimicrobial resistance and gram-negative bacteria that particularly caught my interest, as I was able to learn more about the techniques that I was able to see during my work experience, such as the microbiology techniques used to identify infectious diseases from stool and blood samples, techniques like gram staining, microscopy, and various biochemical tests. After A-levels, I applied to the University of Nottingham to study microbiology. Microbiology is the study of all living organisms that are too small to be seen with the naked eye. These living organisms can include bacteria, fungi, viruses, and many more, which are collectively referred to as microbes or microorganisms. The background image is actually from a gram stain I did, and this is what I could see under the microscope. The initial sample was from a bin outside my university, showing that to the naked eye, the bin looked quite clean, but under a microscope, there was obviously a lot going on. Some topics covered in my degree are food microbiology, which looks at keeping microbes out of our food, but also how we can use some microorganisms to our advantage, for example, corn. Medical microbiology, which looks at diagnostic methods in clinical and hospital settings. Pharmaceutical microbiology, plant microbiology, which covers topics like the effect of microbes on crop production, and biotechnology. The modules that I most enjoyed involved understanding key concepts and methodologies used in the diagnosis of microbial infections, gaining awareness of new and emerging technologies for rapid identification of pathogens, like whole genome sequencing, understanding how bacterial and viral infections are treated, and how pathogens can become resistant to treatment. A key module that stood out to me was microbial biotechnology, which looks at how microbial activities can be exploited in processes ranging from food production, pharmaceutical, to biocontrol of disease-causing organisms. For example, the microbiological production of therapeutic products like viral vaccines using DNA vectors. The degree I am on is a 3-year degree that allows you to take a year out after your second year to do either a year in industry or to study abroad, meaning you will come back after your year out to complete your final year of study, making your course now 4 years in total. This isn't guaranteed and requires you to take the initiative to apply and reach out to the placement team provided by the university to register your interest and utilise their resources. I was lucky enough to secure an industrial placement at GlaxoSmithKline in research and development. I'm in a department of Pharmaceutical Microbiology, which uses microbiological techniques and processes aligned to the control of pharmaceutical dosage forms and looks into development of non-sterile and sterile dosage forms for clinical and commercial supply, which in less fancy terms basically means that it is my job to make sure that the pharmaceutical products that we make aren't contaminated with anything bad. My role involves supporting the pilot plant, which is where the products come from. These products can be tablets, capsules, or dried powder inhaled products, like inhalers, used by people with respiratory problems such as asthma. My day-to-day activities involve me being in the lab, carrying out microbiological tests. For instance, environmental monitoring by collecting water samples or sampling the environment itself, such as the equipment, to make sure it isn't contaminated. From these samples, I will carry out various tests like gram staining, motility tests, and biochemical tests to identify any microbes that may be present. I also have projects that involve method validation and testing, which is looking at finding more efficient ways to carry out these tests in line with our business policies. I can also be found at my desk writing lab reports and researching new equipment in order to streamline and make the processes that we currently use more efficient. Finally, I want to give 3 pieces of advice that I think are helpful for all STEM careers, in my opinion. Firstly, all work experience is good work experience. Don't think that you have to spend a week working in a hospital, lab, or a pharmacy in order to demonstrate your competency. You can get the desired transferable skills from any part-time job. From my own experience, I've had part-time jobs at John Lewis, volunteering at a charity shop, and a St. Jude's petrol station. Now, these jobs don't have anything to do with microbiology, but it demonstrates that I have the ability to work in a professional setting, have good communication skills, time management, organisation, and teamwork, which are skills required for any job. So try not to feel put out if your CV doesn't specifically say you had work experience in an area related to what you want a future career in, because you can still show off your abilities. Second is to stay up to date on current events in your area of interest. Being able to talk about the area of STEM that interests you is brilliant, but being able to talk about its relevancy in today's job market highlights even more your level of interest. This ties in with my final piece of advice, which is to use social media to your advantage. Following social media accounts that are related to STEM allows you to stay up to date with current events without having to go out and specifically research them if that is something you don't necessarily find the time to do. But if you're scrolling down your newsfeed and you already follow those types of accounts, the information will naturally show up. It is also a great way to find out about possible opportunities, as these accounts sometimes advertise work experience, conferences, or events that you might find interesting. And you never know, you could come across an event in an area you never knew about that might spark your interest even more. To conclude, I want to say thank you for listening and good luck in your journey.